Welcome to this explainer. Right now, at this very moment, there is a literal high-stakes biological drama unfolding inside you. Every single second, a breathless race is happening in your veins just to keep oxygen flowing to the tissues that desperately need it to survive. The absolute heroes of this story are two incredible proteins, myoglobin and hemoglobin. Today, we're going to decode exactly how their amazing molecular architecture makes our very existence possible.
All right, let's dive into this. Here is our roadmap for today. We'll be looking at 1. The oxygen transport problem. 2. Heme, the molecular solution. 3. Myoglobin, the storage tank. 4. Hemoglobin, the smart transporter. 5. Regulating the oxygen supply. And finally, 6. When hemoglobin fails.
Section 1. The oxygen transport problem, a life-or-death biological hurdle.
To frame our entire explainer today, we really need to answer this fundamental logistical mystery. How does oxygen travel deep into our tissues when it is poorly soluble in blood and, well, diffuses poorly over small distances? You see, basic gas diffusion works totally fine for tiny, single-celled organisms. But for massive creatures like us, oxygen simply won't dissolve enough in our watery blood to keep us alive.
And compounding this whole logistical nightmare is a strict biochemical limitation. Proteins, as you probably know, are built from amino acids. But here's the catch. Absolutely none of the standard amino acid side chains have the chemical capability to reversibly bind oxygen. It is a massive physiological problem. To get around this roadblock, nature literally had to invent an entirely new non-amino acid mechanism to grab hold of oxygen. It demanded a truly elegant chemical solution.
Section 2. Heme, the molecular solution, nature's ironclad carrier.
So the crucial point is getting our terminology straight before we dive into the machinery. First, we have ligands, which is just any molecule that binds transiently and reversibly to a protein. For us today, oxygen is the ultimate ligand. But to hold that ligand, our proteins use what's called a prosthetic group. This is a permanently associated non-protein compound that is absolutely essential for the protein's function. And the star prosthetic group of our story, that's heme.
Now, because heme prosthetic groups contain transition metals like iron, they actually exhibit a characteristic incredibly strong absorption of visible light. What you're seeing in this graph is how scientists can actually detect the different oxidation states of these iron-containing groups. It's a really vivid visual representation of just how dynamic and active these electron-carrying molecules really are within our cells.
Let's break down the elegant chemical design of this heme group. Its core structure is a complex organic ring called protoporphyrin. Right smack in the center sits a single iron atom. Now, this iron must be in the ferrous, or Fe2+ state, because that is literally the only state that can reversibly bind oxygen. This iron atom forms 6 coordination bonds. 4 are flat along the ring, and 2 stick straight out, perpendicular. One of those perpendicular bonds is firmly anchored to the protein by a residue called the proximal histidine. The other? It's left open, just waiting to grab some oxygen. But here is the critical reality, the real so what. Iron easily oxidizes into a useless Fe3+ state. To prevent this disaster, the surrounding protein creates a deep hydrophobic pocket to protect and sequester the heme. Without that protective pocket, the iron would instantly oxidize and, well, we would suffocate.
Section 3. Myoglobin, the storage tank, your muscle's local reserve.
Myoglobin is pretty straightforward, but it's vital. It's a compact monomer made up of exactly 153 amino acids, which wrap around a single heme group into what we call a globin fold. Its main job actually isn't transport. Its job is storage. It has an extremely high, unyielding, constant affinity for oxygen. Think of it as a localized emergency reserve tank sitting right inside your muscle tissues. It eagerly grabs onto oxygen and stubbornly refuses to let go until the intense demands of, say, heavy exercise cause your local oxygen levels to totally plummet.
And this brilliantly illustrates the pure precision of protein architecture. Myoglobin has a remarkably low partial pressure requirement, just 0.26 kilopascals, to reach half saturation. This creates a very simple hyperbolic binding curve. But the true genius here is the distal histidine, or His-E7. This specific amino acid sits right near the oxygen binding site and acts like a structural gate. It forms a stabilizing hydrogen bond with the incoming oxygen. Why is this a lifesaver? Because free heme actually prefers carbon monoxide to oxygen by a terrifying factor of 20,000 to 1. Just think about that. But by acting as a physical gate and selectively stabilizing oxygen, his E7 drops that dangerous preference all the way down to just 40-1. This microscopic architectural detail actively prevents ambient carbon monoxide from poisoning us every single day.
Section 4. Hemoglobin, the smart transporter, basically shape-shifting delivery trucks.
Let's move to the main event and see how this builds. When comparing myoglobin to hemoglobin, we see the leap from a simple monomer with one heme to a massive, complex tetramer. Hemoglobin is made of 2 alpha and 2 beta chains, housing 4 heme groups in total. Because its whole job is to circulate in the blood, it simply can't afford to have a rigid, constant high affinity like myoglobin's hyperbolic curve. Instead, it features a variable affinity with a very distinct sigmoidal or S-shaped curve. It has to be smart enough to grab oxygen tightly in the lungs, but be willing to drop it off when it reaches your tissues.
It actually achieves this variable delivery through some absolute structural magic known as allostery. Hemoglobin effectively shapeshifts between 2 states. The T, or 10 state, is stable when oxygen is absent. Notice how it's structurally clamped down and stabilized by numerous physical ion pairs, which are also known as salt bridges. This state has a low affinity for oxygen. It really wants to drop it. But when oxygen binds, it literally breaks those salt bridges, shifting the whole molecule into the R, or relaxed state. This new structure is highly flexible and suddenly possesses a massive affinity for oxygen.
The way it actually moves between these states is through a mechanism called cooperative binding. It is an incredibly elegant physical mechanism. Imagine hemoglobin as a row of 4 interconnected mechanical switches. Binding the very first oxygen molecule to a stubborn, clamped-down T-state subunit is super difficult. But when it finally binds, it triggers a physical structural shift across the entire complex. This movement makes it mechanically easier for the next subunit to flip to the high-affinity R state, and even easier for the next one after that. This positive cooperativity is exactly what creates that S-shaped curve, turning it into a perfectly sensitive delivery vehicle.
Section 5. Regulating the oxygen supply. Hacking the affinity. This brings us to heterotropic allosteric modulation. Now, don't let the heavy terminology throw you off. Simply put, this just means molecules other than oxygen can bind to hemoglobin and dynamically influence its primary function. Basically, hemoglobin has multiple external volume dials on its surface. The chemical environment of your blood can actively turn these dials to increase or decrease how tightly it holds onto oxygen. A massive, everyday physiological example of this is the Bohr effect. Think of this as a biological domino effect. Step 1. Your active tissues, maybe you're out for a run, they produce protons, basically acid, and carbon dioxide. Step 2. These molecules physically attach to hemoglobin. The acid protonates specific histidine residues, while the CO2 forms carbamates right on the protein chains. Step 3. This physical attachment creates new salt bridges that effectively lock hemoglobin back into that low-affinity T state. The incredible result in Step 4 is that hemoglobin dumps its oxygen payload exactly in the tissues that are working the hardest. Finally, in Step 5, when the blood cycles back to your lungs, you exhale that CO2, the pH rises, the salt bridges break, and the high-affinity R state is favored all over again, so you can take a deep breath and restart the cycle. It's beautiful. Now, what's really fascinating here is another crucial modulator called BPG. BPG is a highly negative molecule that wedges itself directly into the central cavity of the T state, firmly locking it down to lower oxygen affinity. So, what does this actually mean for you? Well, if you climb a mountain, the air is thinner, right? Your body adapts by rapidly increasing BPG levels. This forces your hemoglobin to drop a much larger percentage of its oxygen into your tissues to compensate and keep you going. BPG is also the absolute key to fetal development. Fetal hemoglobin replaces its beta chains with gamma chains. This tiny structural tweak means it binds BPG very poorly. Because of that, the fetus's blood has a significantly higher oxygen affinity than the mother's, creating a powerful gradient that literally pulls oxygen across the placenta.
Section 6. When hemoglobin fails. The molecular tragedies.
250. Remember earlier when we talked about the distal histidine protecting us from carbon monoxide? Well, even with that amazing protection, carbon monoxide still outcompetes oxygen for the heme iron site by a staggering factor of 250. When CO binds to even just one subunit of hemoglobin, it triggers a catastrophic allosteric lock. It forces the remaining 3 subunits into the high-affinity R state. The hemoglobin holds on to whatever little oxygen it has left with a literal death grip, entirely refusing to release it into the tissues. The result is rapid, severe suffocation on a cellular level.
But tragically, failure can also come from within. Sickle cell anemia traces a devastating pathology back to just one structural error. Step 1. A genetic mutation replaces just one negatively charged glutamate with a nonpolar valine at position 6 on the beta chain. Out of hundreds of amino acids, just one is wrong. Step 2. This tiny error creates a sticky hydrophobic patch that is exposed only when the protein shifts into the deoxygenated T state. Step 3. These sticky patches cause hemoglobin molecules to aggregate and stack into long, rigid, insoluble fibers right inside the red blood cell. Step 4. This internal structural damage distorts the entire cell into a fragile, characteristic sickle shape. Ultimately, in Step 5, these misshapen cells jam up inside your microscopic capillaries. This single amino acid change cascades into extreme systemic pain, severe organ damage, and chronic anemia.
As we see summarized perfectly in this quote, even minor alterations to the structure, as demonstrated by the single amino acid change in sickle cell anemia, can have profound and devastating consequences on health. The entire robustness of your everyday life balances on the impossibly precise shape-shifting of these magnificent microscopic proteins. It really makes you wonder, doesn't it? What other invisible molecular tightropes is your biology walking right at this very second just to keep you alive? Thank you so much for joining me to explore the vital mechanics of myoglobin and hemoglobin. Keep wondering, keep learning, and I'll catch you in the next explainer.